Dear student, I am Dr. Nidhi Sharma, Associate Professor, p.g.govt.college, Jhalawar. In this part of the gene structure, I shall tell you about the eukaryotic gene structure. The eukaryotic gene structure is somewhat different from the prokaryotic gene structure. The eukaryotic gene structure has the following parts: exons, introns, promoter sequences, terminator sequences, upstream sequences, downstream sequences, enhancers and silencers, signals. Upstream sequence signal for addition of cap. Downstream sequences signal for addition of poly A tail. Now about the exons. Exons are actually the coding sequences transcribed and translated into proteins. Coding for amino acids in the polypeptide chain. The exons vary in number, sequence, and length. A gene starts and ends with exons. The exons are from the 5' to the 3' of the DNA. Some exons include untranslated regions, that is, UTR region. Now we come to introns. Introns are coding sequences and are separated by non-coding sequences called introns. Any nucleotide sequence that are removed when the primary transcript is processed to give the mature RNA and functional RNA are called introns. All introns share the base sequence G and T in the 5' end and AG in the 3' end. Introns were first discovered in 1977 independently by Philip Sharp and Richard Roberts. Here is a diagram of the gene in a eukaryotic gene. Here, the black portion is the untranslated region or the UTR region. The red sequence is the coding sequence, that is the CDS, and the blue sequence is the intron. So you can see that there is an exon, then there is a blue colored intron, then again you have got the red colored exon, then a long blue colored intron, then again an exon, and the blue colored gray colored or black colored UTR region. And this eukaryotic gene is the orientation of this eukaryotic gene is from the 5' end to the 3' end. Now we come to the actual process of transcription. Here you can see RNA, DNA primer and the promoter region, which is green in color, and then you can see a dark orange, light orange, dark orange, light orange, and again dark orange region of the RNA coding sequence. Then you have the pink colored sequence of the DNA, which is actually the terminator. Now this DNA is transcribed to the mRNA And in this case of a eukaryotic gene, it is a pre-mRNA. Here you can see a yellow cap at the 5' end. Then you can see the leader sequence, which is brown in color. Then you have got the exon, intron, exon, intron, exon. The last you have got a trailer region, and then you have got the poly A trail with all the adenosine bases at the 3' end. Now this pre-mRNA will be processed and introns will be removed and it will become a functional mRNA. In the next line, you can see that the leader sequence, at the 5' end, you have got the cap, which is yellow and circular. Then you've got the leader sequence and all the introns have been removed from this sequence and all the exons have been spliced in one single gene after the removal of the introns. And then last you can see the trail and the poly A tail at the 3' end. Now the next process is of translation, that is mRNA is changed to a protein or a polypeptide by the process of translation, and you can see this blue colored ring is the result of translation. Now in this case, in the eukaryotic case, a promoter is a regulatory region of DNA located upstream controlling gene expression. The core promoter is a transcription start site -34 upstream binding site for RNA polymerase required for transcription. Then the general transcription factor binding sites. Second is the proximal promoter containing primary regulatory element approximately -250 base pair sequence specific transcription binding of transcription factor binding sites. Now we have got the TATA box. This is also known as the Hogness box, which is upstream -30 to -80 base pairs, and the CAAT, which is also upstream, that is the TATA box, are two distinct sequences. These together are responsible for binding of RNA polymerase II, which is responsible for transcription. Here you can see around 200 base pairs. Then you have got the green guanosine box. Then you have got the yellow CCAAT box. Then you have 100 base pairs. Then you have got the TATA, and then you go upstream, -30 base pairs, and then the messenger RNA continues. Upstream is the 5' end. 5' UTR site serves several functions including mRNA transport and initiation of translation. Signal for addition of cap. The cap is actually 7-methylguanosine to the 5' end of the mRNA. The cap facilitates the initiation of translation and the cap stabilizes the mRNA chain. Now we come to the downstream part, which is at the 3' end and has 3 UTR sites. This downstream part serves to add mRNA, makes the mRNA stable, and it is the attachment site for the polyadenosine or poly A tail. The translation termination codon is TAA. Now, the AATAA sequence signal for addition of a poly A tail is required. Now, here you can see in this diagram, you can see the eukaryotic gene structure. Half of it is on the upstream side and half of it is on the downstream side. The upstream side has firstly the promoter region, which has the CAAT box and TATA box. Then you have got the transcription initiation site shown in pink color. Then you have got the translation initiation codon ATG. Then you have the red colored coding sequences, which are the exons. In this case, you can see 1, 2, and 3 red boxes, which are 3 exons. Now, the introns are the non-coding sequences and which are spliced and removed from a functional mRNA chain. Now, at the end, when you come to the downstream portion, this transcription is terminated, polyadenylation signal starts, and poly A chain is added. Now the whole process you can see in the bottom of the square begins from the 5' end to the 3' end. In eukaryotics, the terminator is recognized by RNA polymerase as a signal to stop transcription. The enhancer enhances the transcription of a gene up to few thousand base pairs upstream, and the silencers reduce or shut off the expression of a nearby gene. Now, what is the significance of introns? We know that introns do not form the part of a functional gene, but still they are present because there is some significance to the presence of introns. Number one, introns don't specify the synthesis of proteins but have other important cellular activities. Many introns encode RNAs that are major regulators of gene expression. Introns contain regulatory sequences that control transcription and mRNA processing. Introns allow exons to be joined in different combinations, which is called alternative splicing, resulting in the synthesis of different proteins from the same gene. Important role of introns is seen in evolution. They facilitate recombination between exons of different genes, that is, exon shuffling. Now, here you can see the alternative native splicing that is done by intron and which is depicted in this diagram, and this diagram shows how alternative splicing is done by the same DNA or the same gene, and this shows the significance of introns. You can see one DNA molecule or one gene which has got exon 1, exon 2, exon 3 and exon 4 interspersed by the blue colored introns. Now, when splicing is done, that is, the introns are removed, exon 1, exon 2, exon 3 and exon 4 remain. Now, in this case, What happens in the third case? Exon 1 is attached to exon 2, is attached to exon 3, and this makes protein A after translation. And in the second case, once again exon 1 is spliced with exon 2 and exon 4. Exon 3 is not spliced in this case, and protein B is formed from the same gene. This is how introns are very, very significant. This finishes our This finishes our gene structure. Thank you.